Good morning. You might think that it's very easy to introduce Professor Pam Soltis because everybody knows her. But the fact is that she has done so many important things over her career that it's actually complicated to give a complete picture of her incredibly rich profile. Pam Soltis is a distinguished professor and curator in the Florida Museum of Natural History at the University of Florida, and is director of the University of Florida Biodiversity Institute. She also serves as Director of Research for iDigBio, the NSF-sponsored National Coordinating Center for Biodiversity Collections. She has served as President of the Botanical Society of America, the Society of Systematic Biologists, the American Society of Plant Taxonomists, and is currently Vice President of the Academy of Science, Engineering, and Medicine of Florida. Pam Soltis received her PhD from the University of Kansas. Her main research interests are angiosperm phylogeny, polyploidy, and the use of digitized natural history collections in biodiversity research. She is widely recognized for her groundbreaking research on polyploidy and has significantly advanced our understanding of flora, of how plants evolve and respond to environmental changes. Working with colleagues, she has made fundamental contribution to our understanding of angiosperm evolution, phylogeny, and plant diversity. She has used these phylogenetic underpinnings to address patterns of genome evolution and ancient polyploidy. Her work on digitized natural history collections, her topic today, has addressed novel uses of herbarium specimens in studies of plant diversity, including niche evolution, and phenological analysis using machine learning and images of herbarium specimens. Among other recognitions, she is a member of the US National Academy of Science and a fellow of the American Academy of Arts and Sciences. Awards include the Dahlgren Prize, the Asa Gray Award, the Stebbins Medal, the Botanical Society of America's Merit Award, and the Darwin Wallace Medal, all jointly with Doug Soltis. Her contributions to editorial boards of journals such as Evolution and Systematic Biology are just two examples of her dedication to the scientific community. But in fact, I also want to point out to her human values, which, as you know, are not necessarily the rule among brilliant scientists. Values that anyone who has interacted with her could attest to, and which are also attested by such recognitions as the University of Florida Teacher Scholar of the Year and Teaching and Mentoring Awards from Washington State University and the University of Florida. Also, to her focus on communicating science to the public and promoting diversity to in the scientific workforce. In all of her roles, she has emphasized scientific literacy, diversity, and opportunities for students and early career scientists. And I should stop now because otherwise I would take all her time. So it's my sincere honor and a huge pleasure to introduce Pam Soltis for the plenary talk today, The Changing Face of Herbarium Collections.
Well, thank you very much, Gonzalo, for that very kind introduction, and thank you also so much for the opportunity and invitation to present some of our work here this morning. And also, I'd like to say a big congratulations to you and your team on an absolutely fantastic IBC. We're enjoying it so much, so thank you very much. Let's see. All right. Oops, there we go. So in considering my topic for this morning's presentation, I thought about how herbaria connect so many of us, maybe all of us, in different areas of botany, even though perhaps we don't necessarily recognize this. So many of us in this room have direct ties to herbaria, but some of us have much more indirect ties. And so I thought it would be fun for the next hour or so to celebrate herbaria and their contributions over the centuries, and also their ongoing and future roles. So to put herbaria into perspective, there are about 3,600 of them worldwide, according to Barber Tiers and Index Herbariorum. And collectively, these herbaria house nearly 400 million specimens. Now, associated with these specimens, though, in herbaria are not just the specimens, but also the libraries, the maps, and all of the other resources, including expert staff, that go along with herbaria. Now, herbaria are really our best resource about what plants occur where and when they occurred there. And so here's a search of the locations for the red maple that I conducted through the portal of IDIG-Bio. And IDIG-Bio is the US National Center for Digitization of Biodiversity Collections. Here I received the reports back indicating these particular locations. And what you can see is that most of these locations map to eastern North America, which is the native range of the red maple. But in addition, there are a few samples that map to other locations. For example, some in western North America. And through a little bit of digging, we identified that many of these locations in western North America are actually botanical gardens or parks, and not misidentifications of species or specimens. But what I think is important here is that natural history collections, including herbaria, really provide us with the greatest resource for understanding and documenting biodiversity on our planet. And as a result of this, we need to really be paying attention to how our herbaria and natural history collections are maintained, how they're curated, and how they're supported. Because again, they really provide us with this foundational information about biodiversity. Now, beyond documenting simply what occurs where, our collections can also tell us about how we know what we know. And this, for example, could come through documentation of particular collecting events. For example, Darwin's voyage of the Beagle. And these specimens, which are shown in the center, are actually housed at the Cambridge University Herbarium, and these represent the contributions that Darwin's collections made to our understanding of plant diversity. Now, for centuries, herbaria have served as the foundation for taxonomy, and certainly this very critical role continues today and hopefully well into the future. But the uses of herbarium specimens actually extend far beyond the foundation of systematics and taxonomy, and I'd like to explore these roles today. So in 2020, a committee established by the US National Academies of Science published its report addressing the roles and needs of biological collections in research and education in the 21st century. I was fortunate to have been invited to participate on this committee, and I had the good fortune of serving with an amazing group of scientists and educators. Now, one of the important themes that runs throughout this report is the fact that specimens have been repurposed over the decades and the centuries, from their initial role of documenting what occurred where to, in some cases, many other sorts of applications, things that couldn't have been dreamed of by the collectors themselves. So who would have ever thought, back when specimens a couple of hundred years ago were collected, that these specimens might be used for DNA sources, and that these DNA sources might form the foundations of phylogenies that include thousands of species? Well, of course, because DNA hadn't yet been identified as the hereditary material, and phylogenetic methodologies were not yet established. Of course, these are applications that far exceeded the potential uses that anyone could have envisioned at the time of collection. So these unanticipated uses, which now extend to areas such as biomimicry and engineering and the bioeconomy, all can contribute to how our collections are used today, in addition to things like systematics, taxonomy, conservation, and the sorts of evolutionary and ecological applications that we currently use our specimens for. And it's these formerly unanticipated uses of herbarium collections that I really want to focus on today. Now, of course, I'm not the only person who's thinking about new uses for herbarium collections, and I think it's been amazing to see through the Congress program so far this week already that there have been symposia that address some of these novel uses of herbarium collections. And so we've seen, particularly on the first day of the conference, several sessions on phylogenomics, many of which use herbarium specimens as their sources of DNA. Yesterday, I think it was yesterday, it's hard to remember, the days are already blurring together, it might have been on Monday. There was really an awesome symposium on herbariomics, again, applications in different herbarium materials in different ways. And then I think also Monday there was a symposium on leaf spectra. And throughout many of the other symposia, there are also talks that have used or are proposing use of herbarium specimens in novel ways. And I'll return to all of these applications at various points throughout my talk this morning. Now, these evolving uses, many of these evolving uses of herbarium specimens are based on digitized records and images. And so I'll be spending a good portion of my talk focusing on what we can do with our digital representations of herbarium specimens, either as text or the herbarium records themselves, or perhaps the images. But we also don't want to exclude the physical specimens themselves. And so I'll talk about that a little bit more later as well. So I'd like to share with you a few brief cases of how we and our collaborators are using various sorts of herbarium specimens in our research. But before I begin with these examples, I'd like to return to a point that Sandy Knapp made in her opening presentation during the opening ceremony on Sunday night. Sandy emphasized how important it is that botanical information be used to address some of our crucial societal problems, because it is botanical information and people who understand this botanical information who may have some important insight into a full range of societal challenges. And so I've listed a few of these societal challenges of the 21st century that relate to biodiversity and where botanical information, such as the information we can glean from herbarium specimens, may be useful. And so most of my examples today actually address at least one, in some cases more, of these particular challenges. For example, challenges associated with climate change, with loss of biodiversity, with food security, and so on. So I'd like to start from this particular starting point, which is the Global Biodiversity Information Facility, which is a global repository of digitized biodiversity occurrences, which also provides us with tools and information to access and use these data. Now, GBIF actually represents sort of the culmination of a global network of other aggregators. And these other aggregators may have been established for regional or national needs. They may represent different taxonomic biases, but there's a whole network of smaller aggregators that sort of feed into GBIF. And all of these can be accessed for information about natural history collections and, in our particular case, herbarium collections. And the result of all of this is really this fantastic network of digitized natural history collection information. So I'd like to begin just with a simple example that may be familiar to many of you. And this begins with digitized specimen records. So on the left of this slide, you'll see a downloaded table, which represents sort of the output that one could get from accessing iDigBio or GBIF or any of these other sorts of aggregators. And this provides us with information about each of the specimen records that we've queried, including the localities. And these localities then allow us to link an individual specimen with a point on a map. and therefore a set of environmental conditions. So a recently collected specimen might likely have GPS coordinates, and those GPS coordinates would allow us to put that specimen directly on a map. But some older specimens, for example, like this pitcher plant from northern Florida, lacks GPS coordinates, and so we need to do a translation from the textual information into GPS coordinates in a process called georeferencing. And then once we have those GPS coordinates, we can then take that information and put the specimen onto a map. And it goes up here in the Florida Panhandle. So now that we have our specimen on the map, we have the opportunity to derive environmental variation and information that corresponds to that particular location. So we can access other databases, for example, WorldClim, which can give us temperature and precipitation information for that particular location based on those GPS coordinates. We could also access databases for soils, such as soil grids, so that we can also obtain soil information for that particular location. And similarly, other databases and environmental layers can be queried for a particular point, and that information can then be tied to that specimen. And so we can repeat this process for tens or hundreds or maybe thousands of herbarium specimens of a species, and then obtain environmental data that can help us understand something more about the habitat conditions of that species. That we can then take all of this information and use software such as MaxSense software to model species niches and distributions. And this approach then will give us the characteristics of suitable habitat for a species, which we can then project back onto a map, and then maybe also even project it into a map of different environmental conditions, such as the map of the future environment. So this is just an example of how this might work. On the left, you see a photograph of the scrub plum, Prunus geniculata, which occurs in central Florida. And it occurs on the Lake Wales Ridge, a hot, dry region of the state of Florida. We can then build a niche model based on its herbarium collections, and we can identify where the most suitable habitat is. And this is represented in the warmer colors in the map in the center. But we can also take that information about habitat suitability and map that onto a projection of environmental conditions for the future. And when we do that for the conditions that are predicted for Florida for 2050, much hotter and drier, only 25 years from now, we can see through the expansion of those warm colors that suitable habitat will be much more widespread than it is at the present or into the very recent past. Now we can do this for a single species, such as the scrub plum, which requires conservation attention, or we can do it for an entire flora and see how a particular flora might respond to climate change. Now, one of the recent studies that we've conducted has focused on the seed plants of North America. And this particular study did not use these models, but used instead simply the herbarium records to map out the species distributions. So in this particular study, we were interested in the phylogenetic diversity of North American seed plants. And here, phylogenetic diversity refers to the amount of the tree of life that's present in a particular area. So we asked the question, are plant species within grid cells, and in this case the grid cells were 50 kilometers square, more closely or more distantly related to each other than expected by chance? So we queried IDIG-Bio and GBIF and obtained about 11 million specimens for these North American seed plants. We used phylogeny from Smith and Brown, and we used the phylogeny and the locations to construct a map of phylogenetic diversity in these 50 by 50 kilometer grid cells. Now, in this figure, the map on the left shows the amount of redundancy that we obtained for the specimen records, where the darker regions represent, for example, California, represent the areas where there is greatest sampling. In the center, we have a map of phylogenetic diversity. And this phylogenetic diversity shows us that throughout most of North America, the grid cells contain species that are more closely related to each other than expected by chance. Now, there are a lot of different phylogenetic diversity metrics and ways to drill into this pattern in more detail. And one of the alternative metrics that I've illustrated here is the relative phylogenetic diversity, another metric that can help us understand the relationships among the species that occur in close geographic proximity. And what you can see when you look at the figure on the right is that there is a very sharp demarcation in the mid-continent, with the region to the east of that central line representing more distantly related species than expected within grid cells, and to the west, more closely related than expected. Now, these patterns are often interpreted as also representing the ages of those communities. And so this pattern would be consistent with the flora of eastern North America being older and the flora of western North America being younger. Now, the main results from this study are summarized here. Again, there's significant phylogenetic clustering across North America, and we see this difference between eastern North America potentially reflecting this older flora and western North America potentially reflecting the younger flora. We also were able to identify some biodiversity hotspots, and these are located in California, Florida, central Mexico, and a little bit farther south in Mesoamerica. And this is interesting because these are biodiversity hotspots that have been recognized on the basis of many other sorts of data over longer periods of time. And so we were quite satisfied that this analysis would actually allow us to identify these as well. We were also able to identify centers of endemism in Mexico, the southwestern US, and the southeastern US. Again, consistent with other hypotheses that have been raised based on other sorts of information. Now, we began that study as really a pilot study to help us explore whether there are enough genetic data in GenBank to make a tree for North American species, and whether there are enough specimen records in our digital databases to allow us to map their diversity onto very broad scales. And we were quite pleased that we were able to sort of conduct these analyses and that the results seemed potentially meaningful. But we also really identified quite a few gaps. We identified gaps in sequence information and also gaps in digitized specimens. And so we decided to go about expanding on this work and developing an improved approach to this whole problem of the diversity of North American plants. So our group was funded in January of this year to tackle the problem of the North American flora on a broader scale. And our plan is really to use a combination of phylogenies, distributional data, and traits to assess both the risk and the resilience of North American plants. So through a combination of niche modeling for the present and the future and trait mapping, we hope to be able to identify taxa and lineages and even areas that may have high extinction risks, as well as taxa, lineages, and areas that may be resilient to climate change. So of course this work has important implications for conservation, and we'll be working directly with conservation practitioners in the latter phase of this project. Now, another challenge for which Herbarium may be able to help provide solutions is food security. And this is particularly in terms of the need for climate resilient crops. And what I'll describe today is just a small part of the work that Mackenzie Mabry, who's a USDA postdoc fellow in our lab, has been working on, along with a team of undergraduates she has working with her. And Tori Ford, a former undergrad, has contributed to what I'll describe here today. Now, herbaria are clearly underused resources for the study of crop wild relatives, including the identification of the closest relatives of our crop plants to begin with. Now, through the use of herbarium specimens, we can actually create ecological niche models for the close relatives of our crops, and then use this information to help identify those particular crop wild relatives that may, through breeding programs, help make our crop plants more resilient. And so herbaria can provide the foundation for a broad range of studies of crops and their relatives, including the identification of those closest relatives themselves. And we can use digital records to ask a few specific questions. For example, what are the environmental limits of known crop wild relative populations, and where in the world do crop wild relatives have high habitat suitability, particularly outside of their native ranges? We can address these questions through ecological niche modeling. And again, I'd like to return to Sandy's talk from Sunday night and the phylogenetic analysis she showed for eggplant and its relatives. Mackenzie and Tori also tackled eggplant using specimens from crop wild relatives and ecological niche models. And here they identified that Solanum incanum is a species of crop wild relative that might have some of the best potential for being included as a germplasm for future crop breeding of cultivated eggplant. And we can see this because through ecological niche modeling, they were able to identify this region of Eastern Africa as the place with the most highly suitable habitat within its sort of accessible region. But then if we want to think about where this germplasm might be able to provide other benefits, we want to see how that germplasm might do on a global scale. And here we can see also through these dark colors that there's suitable habitat throughout much of the rest of the world. And so we think that Solanum incanum, through these studies and a few others that we're conducting, along with a couple of other species, might make good prospects for future breeding for the cultivated eggplant. Now, another use of specimens is for the analysis of plant traits, or the functional traits that ecologists are interested in. Now, through manual scoring, or perhaps more likely using AI, these plant traits can be extracted from either herbarium specimen images, or in some cases, even the text that is associated with the label, or of course, through textual materials found in herbarium libraries. And then these traits can be included in trait databases such as TRI or Austrates. Now, if you're interested in learning more about specimens and functional traits, I would just point you toward this excellent review article by Mason Heberling, which was published in 2022 in the International Journal of Plant Sciences. Now, a trait that's been getting a lot of attention in recent years because of its relationship to climate change is phenology. Now, phenology refers to the timing of seasonal events such as, in the case of plants, bud burst, flowering, and fruiting. And all of these aspects of plant phenology are being affected by climate change, with, in many cases, plants having bud burst earlier, flowering earlier, maybe flowering longer, fruiting at different points in time, and so on. But how do we actually record if plants are leafing out earlier or flowering earlier, extending their flowering later, and so on, or showing other changes in phenology that might be associated with climate change? Well, for nearly 20 years, people have been using herbarium specimens and their records as ways of marking where and when particular events of phenology have been taking place. And so these herbarium specimens actually can offer us decades, if not centuries, of baseline information that can be used to help document phenological shifts associated with climate change. So specimens actually have two types of data that can be used for this purpose. For the first, there may be information that's found in the label itself, perhaps in the text. So in this specimen of this red maple, we might find that it says something like tree in full fruit or something like that. And that information could be mined and incorporated into a phenological data set. Or the information can be taken directly from the specimen itself through scoring of flowers or fruits on the specimen. So we've been working with a couple of groups of computer scientists over the past several years to develop methods for delimiting reproductive structures, such as flowers and fruits, from herbarium specimens using AI. And in a pilot study, we explored the use of machine learning to identify whether an herbarium specimen had flowers or fruits. And although the algorithm could not always differentiate between flowers and fruits depending on the species, the algorithm was 96% accurate in identifying some sort of reproductive structure on that plant specimen. And so this seems quite promising that with some tweaking of the algorithms and perhaps better training sets, we might, for many groups of species at least, be able to use artificial intelligence and herbarium specimens to measure whether or not a specimen is reproductive, and then associate that with the date on which that specimen was collected. And other groups of researchers are similarly exploring these topics. Now, in this particular case, we used an algorithm that identifies either flowers or fruits as before. And you can see, though, that there might be differences in whether or not a particular approach could work for a specific group of plants. So on the left is the California poppy. And with its single large flowers, it was very easy for the algorithm to say, yes, this is a flower, and this other stem just has a leaf on it, or perhaps maybe there's a fruit. But it was very easy for these reproductive structures to be identified through the algorithm. In the center, again, we have a maple, and here we have actually a combination of both flowers and fruits on the specimen. And the algorithm had some difficulty sometimes determining whether something was a flower or a fruit, but it very clearly could say that this had reproductive material on it. In the specimen on the right, this is an orchid with a tightly packed inflorescence of flowers. The algorithm really did not do well in this particular case of identifying there being flowers or fruits or even reproductive structures at all. So I think you can see that the plant groups and their morphology and their reproductive strategies may actually affect how effective these AI methods might be. But these methods will soon be applicable to much larger sets of herbarium specimen images for the analysis of phenology associated with climate change. Now, phenological data are actually being collected by multiple sources, including citizen scientists or participatory scientists, through expert monitoring of specific regions in phenology networks, as well as through herbarium specimens. And so our colleagues have developed a new database or knowledge base called Phenobase. And this was developed by the team listed here. And it integrates all of this sort of information using ontologies developed by the Plant Phenology Ontology Group. So this will help us better understand what sorts of phenological changes have occurred, what we can expect to see in the future, and will provide a really great resource. It's still under construction, but it should be finished within the next year or so, I think. So I'd like to take a few minutes to share some truly creative science that's being done by members of the community using herbarium specimens. And one of the, I think, really creative examples is illustrated here in some work by Emily Meineke and her collaborators. They developed an AI method to score herbarium specimens for herbivory. Now, of course, as collectors, we always want to get the most pristine individuals to use in our collections to prepare our specimens. But in fact, having specimens that show some degrees of herbivory can actually be super important as well. And if you can use these methods to identify not only whether or not there is herbivory, but the type of herbivory, such as marginal feeding or interior feeding or stippling or leaf mining. All of these sorts of information can be particularly important for understanding the ecological interactions of a particular region. And again, this can be done across space and time. And so the method that Emily and her collaborators developed was quite successful as in this pilot study, and it'll be really great to see what comes from this into the future. So what's next in digitization? Now, a large digitization project that was led by my colleague Dave Blackburn has used CT scanning in understanding attributes of vertebrates. And as defined here, CT scanning is a non-destructive technology that reveals a specimen inside and out, its skeleton, muscles, circulatory and nervous systems, internal organs, parasites, eggs, and stomach contents. And this is taken from the OVERT Thematic Collections Network website. But what about plants? So CT scanning is being used on plants as well. And this shows a set of scans of modern walnuts by my colleague Steve Manchester. And aren't these beautiful? These are amazing scans. But there's more, because these technologies can also be used on fossils. And here we see on the right a plate of Eocene walnuts that have also been subjected to the same sort of CT scanning. And the bottom row here shows the same sort of exquisite preservation as the details from the modern walnuts. So CT scanning certainly holds a lot of potential for plant materials as well. And not just for fossils and for structures such as fruits, but also through some experiments that one of our students, Annika Smith, has begun through rehydrating flowers from herbarium specimens and also applying it to pickled or preserved materials within herbaria. Now, next generation digitization also applies to the capture of leaf spectra, as discussed in a few symposia and talks earlier this week. Now, when light hits leaves, it can be either absorbed or transmitted or reflected, and what happens to those spectra really is dependent on the species, the leaf characteristics, and various sorts of other properties of the plant. So there's a lot of attention from plant ecologists about whether or not these plant spectra can be captured from herbarium specimens, and if so, what would that actually mean for understanding more about the plants themselves. And so ecologists are interested in the leaf and canopy traits, in what we might be able to infer about the level of stress that a plant is under, whether or not we can use this information to identify species, and there was a really nice talk a couple of days ago on that application. And then ultimately, as a way of perhaps incorporating a lot of this information into an integrated phenotype that encompasses all of these aspects. So wouldn't it be great if we could actually gather specimens from a whole set of herbarium, or spectra from a whole set of herbarium specimens, and as a way of capturing data from a particular region in an ecosystem across space and time. And this is something that Janine Cavender-Barris has been working on for several years. I noticed she has a paper that is probably almost 10 years old at this point on this topic. And Janine and Doug Soltis and I have been discussing how we might be able to develop high throughput scanning for spectra so that we could actually capture these as another mechanism of digitization of herbarium specimens. And we think there's a lot of potential, but a lot of testing would need to be done. And so what I'd like to do now is just challenge all of you to think about what your concept of next generation digitization might mean in regards to your own work, and how, whether you're an ecologist or a physiologist or a systematist or whatever aspect of plant science you study, how could herbarium specimens actually provide you with some new information? So as we obtain more and more data from a specimen, from morphology, DNA, to isotopes and other sorts of information, the specimen is no longer just a single pressed plant, but it actually includes these derived materials and data that are scattered across different data sets. So how the digital connections can be made and maintained and then retained for use is something that is really requiring a lot of attention. And we refer to all of this, the pressed specimen plus all of its derivative materials, as the extended specimen. And of course, as we want to be able to access all of this information digitally, it's the digital extended specimen. So really a lot of thought and effort are being focused on this concept of the digital extended specimen. And if you want to learn more, I would just direct you to Jutta Buschbaum's talk on Friday, the Digital Extended Specimen Network from Manual Links to Machine Action Accelerating Network Generation. So I think you'll find this presentation to be really enlightening about the value of connecting all of this information. So far I've focused on digital data obtained from specimens, but for the remainder of my talk, I'd like to actually return to the physical specimens themselves. Now this article by Burbano and Gudeker, which appeared in Science last October, really made a great case for the use of herbarium specimens as sources of DNA. And of course, this wasn't really a surprise to many of us in the field and to probably most of us in this room, but it was great to see this topic covered with such publicity and in such a lovely paper. In addition, this paper also called out the importance of the extended specimen, where a specimen might provide us not only with information directly about the plant, but maybe we could obtain information on the genomes of the microbes or maybe some parasites or associated species that might have hung around right there on that specimen. Also, we could obtain things like isotopes and other sorts of information. So my final example of applications of herbarium specimens uses physical specimens as sources of DNA for a large phylogenomics project. Now, I'm part of a team that has used DNA from herbarium specimens to understand one of the most important biological processes on our planet, and that is the conversion of atmospheric nitrogen into nitrogen that is usable by organisms. And this process of nitrogen fixation takes place through a symbiotic association between bacteria and the root nodules that certain plants form that encase those bacteria. Now, the most familiar plants that have this sort of symbiosis are the legumes, that is the bean and pea family, but it's actually more widespread than simply the legumes. And so our goal was to understand the evolutionary origins of this process and ultimately apply what we can learn about the process to bioengineering bioenergy crops such as poplar. And this work is actually funded by the US Department of Energy. Now this diagram will look familiar to some of you, maybe many of you, but many of you of a certain age, maybe we'll say. This is Cronquist's famous bubble diagram for the so-called dicot part of the angiosperm classification. And this is important because it's long been known that 10 families of angiosperms have these nodular nitrogen-fixing symbioses with bacteria. And based on Cronquist's classification, these nitrogen-fixing families were considered to be distantly related, with representatives falling in these 4 subclasses that have the red arrows. And this suggested that there had been multiple origins of this root nodule symbiosis, and this implied to agriculturalists that it might be pretty easy to transfer this sort of symbiotic relationship into non-nodulating crops. But then a big surprise happened. Now, as part of the broader community's sequencing of angiosperms and phylogenetic analyses based on the chloroplast gene RBCL, we discovered in the early 1990s that, in fact, these 10 families were not distantly related, but in fact, they all formed a relatively small clade, a clade of about 30,000 species, but still for angiosperms, not too big. But they all formed a single species along with some non-nodulating species, all within the rosids. And they all occur, this clade occurs in this rosid 1 clade. So instead of being spread throughout angiosperm phylogeny, all of those nitrogen-fixing families form a clade within this rosid-1 clade. Now what this suggested, because there are also species that lack the nodulation, is that there is a single origin for what we called the predisposition for symbiotic nitrogen fixation. And following that origin of predisposition, whatever that might mean in a biological sense, there were probably multiple gains and losses of that trait to account for the distribution within that clade. Now, this was perplexing to biologists, as you can imagine, and it's particularly perplexing still because genomic studies suggest a single origin followed by many repeated losses of this nodulation syndrome. So our team decided to tackle this with a large phylogenetic analysis, and this just represents our team, and I'd particularly like to call out Heather Rose Cates, who was a postdoc on the project, and Ryan Folk from Mississippi State University, who was also instrumental in the success of this project. Now, our goal was to reconstruct the phylogeny of the nitrogen-fixing clade, again, this clade of about 30,000 species, using a sampling strategy that included about half of those samples. So our goal was 15,000 samples, and where do you, in a short period of time, get access to 15,000 different species of rosids that are distributed around the world? Well, you go to your local herbarium. And so we went to our local herbaria, but we also went to a bunch of other herbaria, and we obtained 15,000 samples, primarily from the New York Botanical Garden and the Missouri Botanical Garden. But many, many thanks to all of the herbaria that provided us with these resources. We then used a target capture approach with some probes that Ryan developed specifically for this clade, and we generated a phylogenetic tree and mapped onto it the nitrogen fixing symbiosis based on evidence from nodules that had been actually physically detected on species. And this was our result, published just a couple months ago, where we found that the pattern remains complex, but maybe we have a slightly better handle on this complexity today and on some of the processes that are involved. So we'll dig into this just slightly. Now, one of the things that happens with big phylogenies is that big phylogenies such as this are susceptible to oversimplification during such processes as ancestral state reconstruction. And so we didn't use just a straightforward single mapping of the trait across the tree. Instead, we used a so-called hidden state method that allows us to avoid some of these artifacts of oversimplification. And through this process, we identified that the most recent common ancestor of the nitrogen-fixing clade lacked this nodulation state and was in a so-called precursor state. We then found that the gain of nodulation is actually a two-step process. You have to go from the precursor state to an intermediate hidden state and then onto the nodulation state. Now, of course, we don't exactly know what these precursor or intermediate states are in a biological sense, but from a statistical sense, these are important elements of the model. And this particular model is so much better than the next closest model that could possibly explain the distribution of these characteristics. So using this approach, we then identified 16 gains and 10 losses of nodulation across this clade. Now so we have a good understanding of these evolutionary patterns, but we still need to reconcile these patterns with the genetic mechanisms that may be responsible. And this includes trying to understand what this precursor or predisposition state might actually be in terms of biology. Now, this actually just points toward a lot of additional research that needs to be done, but all of this could not have been possible without the contributions of the herbaria and the generosity of so many herbaria in allowing us to sample from their specimens. So this morning, I've shared some examples of how digital records, images, and physical specimens can bring new biological insight to a range of problems, including some of today's biggest societal problems and challenges. But there are also many challenges that are facing today's herbaria, and I've listed just a few. But these few range from technical challenges to personnel challenges, space challenges, to how we develop possibilities for continued and responsible growth of our collections. And all of these essentially relate to funding and the fact that most of our collections are insufficiently funded, with threats of closures and losses of specimens. In addition, there are issues associated with the colonial legacy of our collections and our desire or need in many cases to repatriate specimens either digitally or in terms of the physical specimens. So I'd like to close with a call for global collaboration in support of herbaria, given the fact that these herbarium collections actually touch on nearly all aspects of plant biology, even if it's only because you actually know the name of the plant that you're working on. I've focused so far on research applications, but herbaria are also important centers for training and also for outreach. And all of these aspects of herbaria need to be supported and strengthened. And so I'd like to end by acknowledging all of the many people who contributed to the work that I described here today, especially Doug Soltis, who's been involved in just about all of it. And this has definitely been multiple team efforts, so I also want to make a big pitch for the importance of collaboration. I'd also like to acknowledge our funding sources, the U.S. National Science Foundation and the U.S. Department of Energy for funding, and to many herbaria, again, for providing access to their samples and their data. And thank you all very much for your attention.
Thank you so much, Pam, for this really great wide overview on all the unprecedented uses of herbaria. We have around 3 minutes for questions, so any question around? Mutama?
Thank you very much. Pam, that was beautifully presented and eloquently showed the future uses of, current and future uses of the herbaria. Muthama Mwasia from the Bolas Herbarium in South Africa. I've got a question on whether you can share some insights to us as a community on how we can together proceed to that future. As you've mentioned, there's a colonial legacy that there are more specimens in Missouri from Madagascar or from Tanzania. And if you look at the participants from Tanzania, I don't think I've seen any here. How can we avoid propagating that legacy as we go into the future? Thank you.
Thank you very much for that question and for your comments. I think you've hit on some really important points. As we think about the just way to distribute our information and our specimens and our digitized specimens, I think it's really important for us to consider how best to do that, in what context, and of course, doing any of those sorts of projects in collaboration with the people who have been most affected. And of course, one of the big consequences of having the specimens not in the home countries is that the expertise is not developed in those particular areas as well. And so I think a concentrated effort on capacity building and data sharing, potentially repatriation of specimens, if that is what is most appropriate for a particular space, could go a long way. How we actually do that, it just, I think, has to come down to having the right conversations, having goodwill, and mapping out a strategy that we can then work toward together as a community. I don't have any good ideas on how we might actually do that, but I think you're raising this question here is an important first step, so thank you.
One other question, quick question.
There's one, there was one over there. Yeah, I don't know if we have time.
That's a really nice talk. So I'm really amazed that you can done all those analysis with just a specimen. So my question is, you mentioned the DNA. So people can use DNA to do some genotyping or scheme sequencing. So what is there any like a pilot project going on, or how would you, imagine like if you have all the sequencing data for specimen collection, what kind of analysis you can done with them?
Yeah. So one of the things that was very surprising to us through that large-scale herbarium sampling and DNA extraction was that many of the specimens that we sampled from looked really terrible. They were black, they were shriveled up, they did not look like they could possibly have usable DNA. And when we got back to the lab and did the extractions, again, a lot of times very low yield, obviously degraded DNA. But for using the target capture approach, it worked perfectly. We just didn't need to do the initial shearing that one might do if you're working with fresh tissue. And so we were able to really use just about all of the specimens that we sampled for further downstream target capture and sequencing. As a sort of byproduct of this study, we produced another paper that focused on, whether the greenness of the specimen was a good indication of whether or not it would yield good DNA. And it turns out that the color wasn't actually a very good predictor. There are a number of other predictors, such as the age, but also even the herbarium conditions under which the collections were stored. And that's a paper that Heather Rose Cates led, and it's published in one of the Frontiers journals in 2021. So it would provide some good, I think, benchmarks and a path for using these sorts of resources. But I will point out, like, you can't get good genomes out of these materials. You can get genes, and especially through target capture, but there are definitely limitations to what these materials can be used for, especially the older ones.
I just want to add one comment that in another project, B10K, 10,000 bird genome project, they also got a specimen from the museum. So in the beginning, the DNA sample were terrible, but they managed to build all the phylogeny from the specimen. I think we can just borrow some of their protocols.
Yeah. Yeah. I mean, we built a tree of 13,000 species, so it worked pretty well.
Thank you so much again. Before I ask you for another applause for Pam, please don't leave the room because I have a few announcements to make before you go to the coffee break. Okay. Thank you.